Hi and welcome to this video. We're going to be running through some multiple choice questions provided by the OCR exam board for OCR A-level biology. Okay, so two ways you can do this. First of all, follow the link and do all the questions first. You'll have to skip past the mark scheme. Don't be tempted. Do the questions first and then come back to this video to see if you worked it out correctly, to what the thought process is, and that sort of thing. Or you can do it kind of with me, so just pause the video every time I get to a new question, attempt it for a minute or so, and then you can see how you did. Okay, so let's get going. So the first question here, aerobic respiration has three stages where reactions occur between free-moving enzymes and substrates. Which table shows the correct location? So this is just a recall question, where do things happen? So glycolysis happens in the cytoplasm, not in the matrix. Link reaction happens in the matrix, so therefore it's B, and let's just check with the Krebs. Yes, that's also in the matrix. So the answer is B. 2. Aerobic respiration is a pathway involving a series of reactions. What is the final reaction in the pathway? It's not that. Formation of carbon dioxide, oxidation of cytochrome. That's just trying to confuse you. It's not that. It could be this one. It could be maybe this one. Now this is the best one, C, the reduction of oxygen to water, because we know that oxygen is the final electron acceptor. So that is the ultimate last step. Synthesis of ATP kind of occurs at the same time, but it's sort of, it's linked to the electron transport chain, but it's not part of those reactions. So I think this series of reaction phrase points us towards C as the answer. Okay, on to the next two questions, 3 and 4. Reactions and processes in cells can be endothermic, requiring an energy supply in the form of ATP. Which reaction is endothermic? So we're basically asking which ones need ATP. The conversion of pyruvate to acetate is not endothermic, that's the link reaction, so it's not that one. The conversion of pyruvate to lactate, that is occurring in anaerobic respiration, and again it does not need ATP, it's not that one. The oxidation of triose phosphate to pyruvate, that's the final stage of glycolysis, that's actually very exothermic, releases ATP. So that means that we get to this one, the polarization of lactic acid, and to be honest with you, I don't even really know what that means, but we just get there by eliminating the other choices. I'll put up a little picture here once I figure out a little bit more about it. Here it is. Oops. So there it is, and now we'll move on to question 4. Okay, 4. Decarboxylase and dehydrogenase enzymes are important in aerobic respiration. Which line of the table shows when each type of enzyme is used? Okay, so decarboxylase, removing CO2. And dehydrogenase is removing nitrogen and therefore, removing hydrogen and therefore forming reduced NAD. So if we have reduced NAD, then that's involved. So in glycolysis, we certainly have dehydrogenase. So this is, true. And we don't actually remove CO2 in glycolysis. So we want one of these two. So we're already narrowed it down to C and D. In the link reaction, we have both. Okay, we have dehydrogenase and decarboxylase. So therefore, we know it's C. But let's just check that Krebs is also, what we think. And yes, they are both present in Krebs. So the answer is C. Moving on. Let's look at questions 5 and 6. 5. Striated muscle, this is stripy or skeletal muscle, contains two types of muscle fibre, red and white fibres. Typical percentages of red fibres are shown. So basically it goes up the more long distance the athlete is. So what can therefore be deduced? Well, if long distance runners have a lot of red fibres, long distance runners need muscles that don't fatigue easily, so it's A or B. And also the waste products are going to be carbon dioxide and water, because this is aerobic and this would be anaerobic. Now, of course, remember in humans, we can't do anaerobic respiration for a long time. It's just a short burst of respiration that generates an oxygen debt to kind of go above and beyond the aerobic capacity for a short time. But marathon runners can't maintain that for a long time, so they need aerobic respiration, so A. 6. Oxygen is required to oxidize food molecules in aerobic respiration. Which food will require the largest volume of oxygen for it to be oxidized completely? We've got 1 gram of all the things. That's just a check, make sure it's kind of control variable. Yeah, 1 gram of each one. And the answer here is going to be lipid. Now, you can kind of get to that in 2 ways. You can think about RQ value. And the RQ value of lipid is around about 0.7, which is the lowest, which means, therefore, that it needs the most oxygen, because remember RQ value is CO2 divided by oxygen. So the more oxygen needed, it's going to have a lower RQ value. Also, lipid, remember the fatty acid chain is just basically carbons and hydrogens and carbons and hydrogens. So there's no oxygens in that chain, so therefore we're going to need to bring in a lot to oxidize. Okay, moving on to the next 2, 7 and 8. Okay, again, RQ. RQ going from 1 to 0.7. So what would this mean? Well, aerobic respiration was replaced by anaerobic. No, that would take us from 1 to like 1.2 or something. It would go up, so that's not it. Anaerobic respiration was replaced by aerobic. Well, if it was started at anaerobic, we'd be like at 1.2-ish, going down to 1, so that's not it. The respiratory substrate changed from carb to lipid. That is correct. That's 1, and then lipid is 0.7. And if it was the other way around, it would be 0.7 to 1. So we can see what each one of these would be, and the answer is C. That's the only one that makes sense. A student calculated the volume of oxygen used by a locust as 1.7 cm cubed in an hour, and it produced 1.3 carbon dioxide in an hour. So the RQ value, you just need to know this, is CO2 produced divided by oxygen consumed in the same time period. So I need to get a calculator out. So therefore for this one, it's going to be 1.3 divided by 1.7. So 1.3 divided by 1.7 equals. So that's going to be 0.76. Now don't just quickly go, okay, 0.7 and make a rounding error. 0.76 rounds up to 0.8, doesn't it? Okay, so it's B. Okay, let's go and do number 9 before we move down. So number 9 is about rotanone. Rotanone is an insecticide that inhibits the electron transport chain. So ETC, that's in the mitochondria, that's part of oxidative phosphorylation, isn't it? So will this affect oxygen consumption? Yes, it will, because it blocks the ETC, and oxygen is the final electron acceptor in the ETC. So it will decrease oxygen consumption. Will it affect lactate concentration? You would expect it to increase it, because if it would cause anaerobic respiration to occur more frequently. So therefore B would increase. And just check that this one makes sense. Krebs cycle activity, yes, we would expect it to decrease, because then that's part of the mitochondria and aerobic respiration. So B is the answer. Okay. Oh, moving on to this question. This is a really lovely question. It's going to take a little bit of working. So here's how you do it. Okay, so first of all, it says the top figure shows the basal metabolic rate as joules of energy used per square meter. Joules of energy per square meter of body surface area for people of different ages. Okay. The second figure shows how height and mass affect a person's surface area. Okay. The question now: how much energy is used in 5 minutes by a 20-year-old female of height 170 and mass 60 kg? Well, okay, the first thing we've got to do is we need to work out, we need to basically look at the 2 graphs and think, well, okay, what information do we have? Here, have we given this? No. We've got the age, and we know the person's female, so maybe we could say 20 and female. So we know I've already worked out some information. In fact, I know that the basal metabolic rate is actually 36 joules per meter squared per second. Okay, that's pretty good. Got some information. What's next? Now I need to figure out how much surface area a person has. So I'm going to change colour because to make it a bit easier. So this person is 170 centimetres tall and mass 60. So if their mass is 60, we can look at this line. And if they're 170, that's there. Okay, so I'm going to zoom in a little bit. And in an exam, you'd be doing this with a ruler. So we need to work along this line. And we need to work along this line, which is at 170. It's that line there. So they intersect right there on the 1.7 meters squared surface area. Now we've got to do some maths. This is joules per meter squared per second. So if we times it by surface area, 1.7, we can get rid of the meters squared. So I'm going to do 36 times 1.7. And that gives me 61.2 joules per second, because now I've got rid of the meter squared. So the question actually asked me for how much energy is used in 5 minutes by a 20-year-old female. So 5 minutes. So 5 minutes is 5 times 60 seconds. So I need to take this number and basically times it by 5 minutes, and times it by 60 seconds. Okay, so times it by 300. So 61.2 times 5 equals times 60 equals, and now I've got my answer, which is 18,360 joules. And there it is. The answer is D. That's one of the hardest 1-mark questions I've ever seen. So well done if you got the answer. Okay, let's move on. Okay, question 11 now. This is a tricky one again. And it's kind of an interesting question. I'm almost not sure if this really should be on the syllabus. From my reading of the specification, it seems a bit tough, but here we go. So, which line of the table shows the substances that ionize to release a hydrogen ion into solution? So things that ionize to release a hydrogen ion, they are mainly carboxylic acid. Okay, so which of these are carboxylic acids is kind of what it's saying. So substance J is glucose. Substance K is going to be pyruvate. And then acetyl-CoA feeds into the Krebs cycle. Difficult to really tell exactly what these two things are, but most likely this is citrate. And this is oxaloacetate, I guess, although we don't know exactly where on the cycle they are, but I presume so. 5 carbon, 6 carbon. Okay, so basically if it ends, okay, here's a quick way of remembering it. If it ends in 8, it's a carboxylic acid of some sort. I'm going to drop in a little picture here for the full biochemical detail if you'd like it. Here it is, to the side. So pyruvate, citrate, and oxaloacetate are all sort of carboxylic acids, and therefore we could say that K, L, and M all will release hydrogen ions in solution. So the answer is C. Glucose won't. So the answer is C. There we go. Moving on. 12. Chemiosmosis produces ATP. What explains ATP synthesis by chemiosmosis? This is really a spot the errors question. When you read the sentence, spot the error. So hydrogens diffuse from the intramembrane space to the stroma through the ATPase. Now, if you just read A and weren't paying attention, you might have missed that and just went, oh yeah, A. The stroma, that is chloroplast. So we're not talking about chloroplast, though we're talking about respiration right now in this question. Yeah, and also, okay, so the reason why it wouldn't be stroma as well is intermembrane space. If it was photosynthesis, it would say from the thylakoid space through to the stroma. Okay, so that those two things kind of work together are wrong. B, hydrogen jump from the inner membrane to the outer membrane. No, that's definitely not true. Hydrogen ions aren't in the membrane. C, hydrogen ions diffuse from the intermembrane space to the matrix through ATP synthase. That is correct, but we always check the last one as well. Protons are pumped from the matrix to the intermembrane space. No, they're definitely not pumped. It's not an active pumping. So C is the answer. 13. Cells in culture were supplied with radioactively labelled oxygen gas O2. Which cell metabolite will show most reactivity after a few minutes? So basically it's saying, where does oxygen get incorporated in respiration? It's needed, but where is it actually incorporated? It's the final electron acceptor. Remember, it's used to take hydrogen ions and produce water. So therefore the answer is D. Moving on. Okay, a bit of mass here. This table compares the oxygen consumption and body mass of two animals. These figures can be used to calculate the metabolic rate of each animal in centimeters cubed per gram per hour. Now, the thing which you really have to do here is we don't really teach that much about this in the syllabus, but the question gives you multiple clues. The clues are given in the units. The metabolic rate of each animal is in centimeters per gram per hour, centimeters per gram per hour. Okay, so how can we work that out? The units here are centimeters per hour, and the units here are grams. So to work out the metabolic rate, you've got to do centimeters cubed per hour divided by the mass. So what the sum that I'm doing to work this out, 52,500 divided by 5 million, and 20 divided by 10. 20 divided by 10 is just 2, no probs there. But what about the other one? 52,500 divided by 5 million equals 0.0105. I'm still not the answer yet, because it asks me how many times greater is the shrew's metabolic rate. How many times greater? How many times greater is 2 versus that? So therefore we do 2 divided by 0.0105. 2 divided by 0.0105. And the answer is 190, which is one of the answers. So it's C. Great. Moving on. Question 15. This is about simple respirometer. So in a simple respirometer, the gauze bag could either be filled with soda lime, which absorbs CO2, or just with glass beads, which would have sort of no effect really. Okay, so can this device be used to measure the rate of oxygen uptake? Yes, if you have the soda lime in there, it will just be measuring the absorption of oxygen. 2, can it be used to measure the difference between oxygen uptake and carbon dioxide production? Yes, it can. If you don't have any soda lime in the gauze bag, just glass beads, then this thing is taking in O2 and releasing CO2, so therefore the change in volume will be down to the difference between those two. Statement 3, it can be used to measure the difference between carbon dioxide uptake and oxygen production. Those two bits there are wrong, aren't they? To spot the errors. Carbon dioxide uptake, no, it's not a plant, it's not photosynthesizing, so therefore it's just 1 and 2. The answer is B. All right, moving on. Zooming out a little. Let's do 16 and 17. Which of the following statements is or are true of respiration under anaerobic conditions in both yeast and animal cells? So just, you might want to quickly remember. Yeast is basically glucose goes to CO2 plus ethanol, and animals is glucose goes to lactic acid. Simple. Okay, so carbon dioxide produced. No, not the same for both, because only that one, so no. Glycolysis occurs. Yes. Okay, glycolysis always occurs, and then the fermentation is the extra step, which is needed to oxidize NAD. So that step is also true. So 2 and 3 are correct, so the answer is 16C. 17. Which of the following reactions form part of glycolysis? Okay, ATP donates a phosphate group to glucose. Yes, that's the very first step of glycolysis, isn't it? Statement 2. Triose phosphate is oxidized to pyruvate. Yes, that's the last step of glycolysis, kind of energy harvest. And statement 3, a 6-carbon sugar bisphosphate splits into 3 carbon sugar phosphates. Yes, that's true. Yeah. Because remember, glycolysis is like glucose. I won't draw it all out, but we take ATP in. Twice, actually. and then we have the hexose bisphosphate, and then we split that into 2, and then we have triose phosphate, and then we oxidize that to pyruvate, and we get energy out. Okay, so that's kind of an overview. So statement 1 is up here, statement 2 is down here, and statement 3 is in the middle. They put them in the wrong order to try and confuse you. So it's 1, 2, and 3. 17a. Right, almost there. 18. Diagram of mitochondria, a bunch of different reactions happening there. Which statements concerning the diagram are true? The acetyl group from acetyl coenzyme A will join with oxaloacetate. Yes, that is true. So remember, in Krebs cycle, we have an acetyl group joins oxaloacetate to make citrate. Sorry, I just realised I wrote 5 carbon there. It's 4 carbon, silly. 4 carbon is oxaloacetate, plus the 2 carbon gives us citrate, which is 6 carbon. The reduction of NAD occurs at the cristae. Sounds true, unless you think about it for a second. It's false because it's the oxidation of NAD that occurs at the cristae. Hydrogen ions are removed. So look, we can see here, this is oxidation. It's happening here. Substance Z is glucose. No, that's not true. It is pyruvate. So it's just number 1. 19. Live yeast is used to make bread. The yeast is mixed with flour, sugar and water to make dough. This is left in a covered bowl for 1 or 2 hours. During this time, the yeast cells respire and produce bubbles of gas, that's CO2. That makes the dough rise. Which of the following statements describes an effective way to shorten the time needed to make the bread dough rise? Well, statement 1, if you heat up the dough, yes, it's going to make the dough warmer. Respiration is going to occur faster. If you watch Bake Off, you'll know that's also true. Statement 2, make the dough with double the normal quantities of flour and water. Not really. Double the sugar, maybe, but just double the quantities of flour and water no, that's not going to do anything. Or with the yeast, no. 3 is a tricky one. And I've debated this with students in the past. Use finely powdered sugar instead of granulated sugar. So this is a distractor. So they want you to go wrong here because this is high surface area, isn't it? High SA versus lower surface area for the granulated. But when you've mixed it all in the dough, effectively, I guess you consider the dough as sort of a solution or it's fully mixed and the sugar's been dissolved in all the dough. So therefore it wouldn't make a difference really. So the answer is just statement 1, which is D. Finally, another weird kind of application style one about something called a hotbed to grow crops. So early on in the spring, farmers or people with allotments, I guess, might make a hotbed. Dug a pit in the soil, fill it with 50 centimetres of animal manure and then some topsoil on top of that. It's covered with a glass cover supported on a wooden frame. So why is it effective at increasing the rate of plant growth? Certainly number 1 is true. It's the decomposition of the manure, the respiration, the exothermic reactions that cause heat. Statement 2, the soil around the pit and the wooden frame acts as a heat insulation. Not really. Soil's not really great insulator, can conduct heat, especially if it's damp. Statement 3, temperature is a limiting factor for plant photosynthesis. I feel like that's a distractor. I mean, that's kind of true, but is it the reason? Okay, so this is why it's tough, because it says which of the following statements explains why. So even though statement 3 is true, does it explain why the hotbed is effective in increasing the rate of plant growth? The exam board says that this is not the right statement. The exam board says that it should just be only 1. And I don't know, this is a difficult one. I could say that I disagree, but I can sort of see that maybe that they're saying, A, this question pack is about respiration, not photosynthesis, so it's not about photosynthesis. And B, I think when the plants are just growing in this hotbed, it's really more about the germination of the seeds in the early growth than the photosynthesis that comes later, okay? So that is the end of that. Go over the questions again, see if you can remember how we got there. Maybe you could print out a new copy and try it again after a few days, after you've forgotten just the answers and you can still remember the working. Hope that was helpful, and I'll see you next time. Bye-bye.